The second lab, the anaerobic lab, right? The data you'll make next week is on power and velocity between jump squats, or it's a plyometric jump, and speed squats. Just a regular squat, you just do it really fast, okay? There are differences in power and velocity during each of those movements depending on the load of the bar, the resistance is being applied, right? So what do we know about the size principle and motor unit recruitment? Yes, we're going to recruit smaller motor units first, right? If we need to produce more force, we recruit more motor units, and they tend to be bigger motor units. Those bigger motor units are bigger because they're innervating more fibers, and also those fibers are bigger, which results in more force, okay? So when you look at data in humans on force production and adaptations to resistance training, way back, they used to call some human fibers type 2B fibers, because when you compared mouse or rat muscle to human muscle, the fastest contracting fibers in rats and in mice were the type 2Bs, not the 2Xs. Mice have 1s, 2As, 2Xs, and 2Bs. So in our muscle, our fastest muscles were what we thought were 2Bs, so that's why they called them 2Bs. But around 2000, they had the Human Genome Project, and they realized that we do not have the genome sequence to produce the 2B protein. Okay, so I say this because when you go through other literature, and maybe you're writing to your discussion in the lab report, you shouldn't be writing type 2B fibers. You should, even if the paper you're referencing says 2B, you should be using the term 2X, because at that point in history, they thought what was a 2B muscle fiber was actually a 2X. Okay, so mice and animals, they have type 2X and 2B. Humans only have 2Xs. Right? For the mice, their fastest fibers are Bs. For humans, our fastest fibers are the Xs. So that's where that comes into play. So if you see on that, just immediately think not 2B, it's they really mean a 2X. Okay? But we can see here, like this is also telling us the fiber types, but instead, see one of their different gray colors, they're actually using differences in pH to change the enzyme kinetics, called phase shifting, on the head of the myosin molecule, the ATPase on the molecule, right? So if you change the pH in a way and then do the histochemistry, you see that, well, this actually stains as white because it alters the myosin ATPase 2A in a way that it appears white under these slides. That pH, that same pH, changes the ones in a way that makes them look darker, right? So it's still doing fiber typing, except the other way we're looking at the actual myosin heavy chain. This, we're looking at the enzyme, the ATPase on the head of myosin, right? So there is a correlation between the ATPase enzyme and the myosin molecule. You're not going to see a 2X ATPase on a myosin heavy chain 1 myosin molecule, because why would you have a 1 myosin heavy chain and a really, really fast contracting ATPase? The proteins aren't going to, they're producing proteins that are consistent with what the myosin heavy chain is, right? So the ATPase would be the same type as the myosin heavy chain molecule, because the myosin has ATPase on it, on the head of the myosin, right? So those are all related, yes?
So type 1s have slower ATPases, right?
Yes.
So does that slower ATPase play a role in why they're like resistant to fatigue at all, or?
I don't believe that's why they're more resistant to fatigue. It does explain why they contract. They go through contraction and relaxation slower, so obviously their speeds are slower, but they are more resistant to fatigue because of their substrate use and patterns and availability of using oxygen, right? They're smaller, usually smaller fibers, so oxygen diffuses better in those fibers, and they have more oxidative enzymes, so they're able to use that oxygen to make ATP a lot better than some of the other fiber types. But it's not necessarily because of the ATPase that they're more resilient to fatigue. That just relates to the speed of shortening, not necessarily endurance capacity, but good question. So one way you can also look at fiber type is relative myosin heavy chain composition. So if you look at this previous slide, if you look, you can actually count how many type 1 fibers are. There's 1, 2, 3, 4, 5, 6, 7, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 1. There's 10 type 1 fibers here, right? And there's how many 2As? 1, 2, 3, 4, 5 2As. How many X's? and 1, 2, 3, 4, 5 more X's, right? So you can literally go in and count the number of fibers of a type and the size of those fibers of a type, right? So when we say this person has 70% type 1 fibers, they do the biopsy, they make a cross section, they literally go in and count hundreds of fibers to make sure that out of these 400 fibers, this many percent of it was 1s and 2As and X's and hybrids, okay? When the other way, how some people try to fiber type is they take that entire sample, they liquefy it, and then they put it through this gel-like substance. Have you heard of gel electrophoresis? STS page. So essentially, it's kind of like, you know, like Jell-O, right? It's like gelatinous and kind of very wiggly. You know, like the Kraft singles, like the little cheese that's kind of wrapped up in package. Imagine you have a substance that's like Jell-O, but it's a thin, porous kind of consistency, like a Kraft singles. And what you actually end up doing is you put the sample in, you put the muscle sample after you've liquefied it. So here's, right, just imagine this is really, really thin. You put the sample in here, there's like a bunch of things. So this has a sample, this is a muscle sample, muscle sample, muscle sample. They're all liquefied. And imagine that this membrane has pores in it. So you think like you go to drain your pasta out of a strainer, or it's called a colander or strainer, right? They have, the bigger the holes, bigger things can fall out, right? Smaller the holes, only smaller things pass through. So this membrane actually has really big holes in the top, but then has really small holes down at the bottom. So that when you liquefy the entire muscle sample and put it in here, when you add an electrical charge to it, it pulls the samples from like magnets, right? It pulls the proteins apart, so the big proteins stay up here and the small proteins get dragged towards the bottom. So this is how you can separate proteins, like literally thousands of proteins in your muscle. You can do like this. You just look for an antibody to find it. But we know that type 2X myosin heavy chain is a slightly bigger protein than myosin heavy chain 2A. We know that type 1 is slightly smaller than those other two. So if you liquefy the entire sample, the 2X's will get caught up here, the 2A's will get caught up here, and the 1s will migrate a little bit further down because they're a little bit smaller. So this can tell you out of this entire muscle sample, this is the percent of the proteins in that sample that's 2X, 2A, and 1. Now if we look at this, do we know someone's actual fiber type percent? No, right? We're not able to actually know someone's fiber type percent because we're not able to actually count the fibers. For example, you have hybrid fibers, yes? You know that you have some fibers that are like 2AX. So this is a muscle fiber that has some myosin heavy chain 2A and some myosin heavy chain 2X. It's got a little bit in the middle, right? On here, we don't know that. If that fiber had both those proteins in it, when you break up that entire cell, the X's stay up there, the A's stay down there, even if they were in the same fiber, right? So this isn't telling us the fiber type percent, it's only telling us the amount of certain types of fibers in an entire sample, but not the specific fiber number. Why am I saying this? Because if you go to talk about training adaptations with power and the influence of fiber type on power and velocity, there are some papers out there where they will say, oh, well, we found that this type of training changed fiber type. If they only did fiber typing like this and didn't do it like this, they can't say that their training caused a change in fiber type percent because they didn't actually count the number of fibers. All they can say is that their training altered fiber type size and maybe percent in some way, but they can't tell you exactly in which way. Because for example, if the 2A's convert, if the 2X's convert to a 2A, right? But the 2A's didn't get any bigger, they just had converted, you're gonna see there's more 2A's there. Not because they have hypertrophy, just because the X is converted into a 2A. Or conversely, what if you really train the 2A's, they get so much bigger, that's gonna look like, oh, you have more 2A's. You didn't, they just got larger, right? So I can't really tell you that. So I'm saying that because there are papers out there, and if you're you should be talking about more physiology in this lab report because it is more anaerobic, the material is kind of more physiologically more relevant, more related to each other versus like this test versus that test. you should be finding more physiology on the fiber type, motor unit recruitment, that kind of stuff to explain it. And there are papers out there that are perfect for explaining the lab, but some use this methodology, and it's not that this is wrong, you have to take what they say in a grain of salt based on what their conclusions are trying to make, okay? Anyway, so if you look at this example, this is a person, if you look at their muscle fiber relative composition, you'd look at this and you'd say, oh my gosh, they have 10% type 1 muscle fiber, and they have like almost no type 2X. Is their muscle all type 2A? No, right? So this is actually a sample from me when I was at Kansas. I was part of a training study on auto regulation. I mean, I was also at a track and field at my undergrad as well, but I was lifting all throughout my PhD. And I was part of this training study, and I squatted roughly around 475. So almost 3 times my body weight, which was almost 500 pounds, and I weighed about 175, 180 pounds, right? So it was quite a bit. But the thing is, we were training very heavy. I had 10 or 15 years of training history and experience. So the fact that it was always resistance training, maybe some genetics, all of the 2X's had converted into a 2A, right? This doesn't mean that I only had 10% of my muscle fibers in my VL were type 1. No, if you actually count my muscle fiber type, I have still probably around 50-55% type 1, right? So they're still there, it's just that due to the training program, it was so heavy, the X's converted to A's, and then the A's got so big that when you do the muscle biopsy, they push all the other fiber types out, and that's just the sample you get, right? So this kind of just goes to show, like, if the muscles grew, if the type 2A's grew in size, and the type 2X's converted, from here, you can't tell if you still have 2X's left. I probably did, right? But I wouldn't be able to do that because I didn't go in and actually count the fibers, right? So this is the limitation in that. And I'm just bringing this up because there are quite a lot of studies that will also do this in their analyses. Many studies do this type and this type in the same study. So if you're looking at a fiber type paper, you want to make sure that you're drawing the interpretations or inferencing the data, or physiologically what's going on. It's better probably to do it on this data, this methodology versus this one. This can still tell you information about the training, but it's telling you about the fiber type and the fiber size at the same time. This can tell you the change in number of the fibers, and it tells you the size change separately as well, right? Because you can do that, right? So just key thing there. Anyhow, when we look at the sarcomere, remember I said that the myosin heavy chain is kind of like, it looks like like a golf club, right? So the myosin's in the middle, you have the heads of the myosin molecule here. This is the head of myosin, this is the heavy chain tail, right? So remember I said the antibody attaches to the handle of that golf club. It says, looks for a sequence here. Oh, this is a type 1 myosin heavy chain, because this is a type 1 and it's different than that location on a type 2A or a type 2X. On the head of myosin, you have an ATPase, which tells how fast it's going to contract. So usually the ATPase is the same type as the myosin heavy chain. That's why you can fiber type either way, because they're like 0.98% correlated. They're highly, highly correlated. But anyway, when we think about the force production in the muscle, actual production of force occurs at the sarcomere, specifically with the binding of actin and myosin, right there. So the sarcomere is the smallest unit, force-producing unit of the muscle, and that's where the force is produced. If you look at the different fiber types, in general, we see that type 2 fibers are going to have around 10 to 20% more force than the type 1s. This is at least partly due to the fact that type 2 fibers are larger than type 1s. So if you have one fiber and then another fiber is bigger, right? That bigger fiber usually produces more force because it's filled with more sarcomeres, right? If you have more sarcomeres, you can produce more force. So when we talk about hypertrophy and bigger fibers produce more force, we're assuming we're talking about myofibrillar hypertrophy. We're adding more myofibrils, we're adding more sarcomeres. Okay, so those larger fibers are producing more force because they have more sarcomeres. But what we notice is that even if you normalize the force based on their size, like say, oh, you're a bigger fiber, right? You produce more force, great, but what if we say this is how big you are and divide that force by it? It's kind of like absolute VO2 versus relative VO2, right? Men usually have higher VO2s because they have larger lungs, hormonal differences between women and, bigger body sizes. But you could have relative VO2 show that, women have higher VO2 max because they're better oxygen uptake relative to their size compared to the other gender, right? So when you adjust the force based on the size, we can see that even if you adjust it, the type 2 fibers still have more force production versus the ones, okay? We also know that the type 2s are going to contract faster and in greater peak forces than the ones as well. So why are they called slow twitch fibers? Yes, they twitch slowly neurologically, right? But if you look at the single fiber, right, the single fiber is called slow twitch because it reaches peak twitch force slower and it relaxes slower compared to the other fiber types, okay? The type 2A myosin heavy chain reaches peak forces faster than the ones and relaxes faster. And then if you look at the 2Xs, they have the fastest and the highest peak forces, and they also relax the fastest. So when we talk about why are they the fastest fibers, it's because they're able to reach peak force and shorten very fast and also relax very fast, right? Whereas the ones take longer to get there and they take longer to relax. So this relates to power, okay? Key point, this is like totally independent of the nervous system, right? So usually we just talked about like, oh, the action potential, the firing rates, the recruitment of the motor units, like all that is true, but this data here is specifically like if you just have a fiber, like specifically a fiber like that. If you took the fiber out of someone, you clamped it, it has force transducers on either side, you put it in a solution and produce an electrical charge, the muscle will contract. If there's ions there and electricity, like an action potential, it will shorten and produce force. So those qualities about the contraction forces and the shortening speeds, that is independent of the motor, the nervous system input, right? So these are inherent qualities of the fiber, of the sarcomere. Does that make sense? Now, we just mentioned the specific force is going to be higher in the type 2, specifically highest in the type 2X, but notice how, yes, type 2 fibers are bigger than the type 1s, but even if you adjust that force based on the size, they're still producing more force relative to their size. And if you couple that with the fact that the 2As and 2Xs shorten faster, that means they're more powerful, okay? What determines the speed of shortening in the muscle fiber? The myosin ATPase, right? So if the 2X fibers are already big and they have an ATPase that makes them contract faster, what does that mean for power? They're going to be more powerful than the other fiber types, right? So it kind of makes sense that the 2Xs are going to be our power fibers or high force, high velocity fibers because they have an ATPase that shortens quickly and the fiber itself is very big, which produces more force. So overall, it is a more powerful fiber type versus a type 1, okay? Which is why we recruit the type 1s during high intensity exercise, but they're not the ones providing the largest amount of work. They're just kind of contracting along with the X is doing all the work, okay? But if we look at this data, if we say, okay, well, yes, the 2s produce more force, even relative to their size, but why is that, right? It's not because they're producing more force just because they're bigger. You take the size out of the equation, they're still producing more force, okay? So what is the force-producing unit of the cell, of the inside the muscle fiber? The sarcomere. So that means that if all else is equal, there must be something about the sarcomeres and how they produce force that's different between the fiber types, okay? This is where we're getting to more specific generation of force between the fiber types, okay? It's not just that the type 2As and 2Xs are bigger, it is actually something about the sarcomeres in those fiber types that also allow them to produce more force compared to the other fiber types. How do we measure fiber size and fiber forces? Well, you take the single muscle fiber out of the muscle. The muscles, when you do a biopsy, they're actually kind of lined up. Like take dried chicken, for example, right? You ever have really dried chicken, cut into it with your fork, and it just kind of shreds, shreds. Anyway, they're kind of like Twizzlers, right? The fibers are kind of like twisted around each other. They're kind of vertical and kind of twisted around. You can actually take tweezers, really crazy. The muscle fibers are such big cells, you can see them visibly with your eyes. And you can be under a small microscope, and it literally in a petri dish, it looks like a piece of lint or a hair just floating in a little solution, right? But that is a long singular muscle cell. But you can see it with your eyes because muscle cells are so big, right? So you can see those muscle fibers, they're in bundles. You can take a tweezer and pull them apart like Twizzlers, right? Into a single cell, put them in a force transducer, add a solution, an electrical impulse, the muscle will shorten, the fiber will shorten. That's how we measure muscle force. And you can also measure the diameter of that single fiber as well. So that's how we get fiber size and fiber force. Make sense? Okay. So these next data come from, well, not this one, but the next one, will kind of exemplify that. If you look at single fibers, this is in mice or in rats, I forget which one, I think it's in rats. But notice how the rats have 1s, 2As, 2Bs, and 2Xs. Notice how the type 1s, their shortening velocities are a lot slower compared to all of the 2s, right? Notice how the fastest velocity fibers in mice are the 2Bs, right? See how much faster they are compared to the 2As and the 2Xs, right? So this is why when we use, 91, when we used to compare human muscle to mice muscle, we would call ours type 2Bs because our fastest fibers were the X's, right? But they performed like the B's did, so that's why we call them B's. But ultimately, mice actually produce the 2B protein is actually structurally different than the 2X, right? We don't produce the 2B protein, we produce 2X. So this is why they're B's and the X's are not the fastest contracting in mice, but they are in ours. Okay, so that's where the distinction comes in. So there's great muscle physiology papers published. Can we use this article? It's published in 1981 or 1985. Great research is great research. We come to know a lot because technology advances, but if it's a good study design, a solid research question, stats and methods were done appropriately, doesn't matter how old that paper is. If it's relevant and it works for your article and it's done the correct way, you can use it, all right? So seminal articles are, really important research doesn't become less relevant the older it gets, and if anything, it becomes even more important because it lays the foundation where future studies can grow and understand the topic better. Anyhow, if you look at the single, so those are the fiber velocities. So we know that the 1s don't fire or don't shorten as fast as the 2s. Now this is a study based in humans. Again, they did muscle biopsies. They looked at single fibers in humans. Okay, now keep in mind this data is just the fiber. There's no input from the nervous system at all. They just literally took the fiber out, separated them, they fiber typed it. Is it a type 1, a 2A, a hybrid, means it's a type 1, 2A, or it's a hybrid 2AX, right? So they typed it that way. And they also look at the size of that single muscle fiber as well. And what do we see? The type 2s, specifically the 2A and the 2AX hybrid, they produced more force than the 1s and the 2A hybrid, okay? Also, the specific force of those 2A and 2X were also greater, right? So they produce more force, but they also produce more force relative to their size, okay? This is it's a scatter plot of the individual of this fiber's size and this fiber's force in a given fiber type. So the circles are the 1s, the white triangles are the 2As, the black and white triangle is the hybrid 2AX, right? So you can see all those scatter plots there. Remember in that first body comp lab, I said here's a scatter plots and then you make a regression line, right? You can take those individual X, Ys and make a slope, right? So that's what happened here. They took within the fiber type, they looked at relative to the size of the fiber and the force of the fiber, and they took all those individual points and converted it into a regression line. And what do we see? This is the type 1 fibers. These are the type 2A and the hybrid 2AX, right? Notice how the 2A and the 2AX almost lie right on top of each other. They're very similar. There's slight deviation in the slopes, but they are almost on top of each other. But what do we see about this graph? If you look at the x-axis, we can see that from 2,000 to 8,000 micrometers, we see that as fibers get bigger, what happens to force? It goes up. So do bigger fibers produce more force? Yes. Do we see that in type 1s and type 2s? Yes. Notice that the slopes are similar, right? So in both fiber types, if the fiber got bigger, it did produce more force. But what do we notice about what's different between the fiber types?
More dense.
The what?
Type 2 is more dense.
Not more dense. Well, it's an interesting term. You're not totally wrong, but you're on the right track. What is it doing, right? So if we look at 6,000 here, right? If we look at a type 1, 2A, and 2AX hybrid, are these fibers the same size?
Yeah.
Yes, right? The fibers that were measured of this size, they were that size, right? So these fibers that are here are just put into the regression and it comes out on the line here, right? So these fibers are the same size. Didn't we just say bigger fibers produce more force? But if these two fiber types are the same size, shouldn't they produce the same amount of force? The sarcomere is different, right? They're the same. Bigger fibers produce more force. But if you look at different fiber types of the same size, they actually produce different forces, right? The 2s, the 2AX and the 2As, at any given fiber size, the 2As or the 2AXs are always producing more force than the 1s. So what does this mean? This means you can have a 1 the same size as a 2, and even though they're the same size, the 2s are always going to produce more force. Right? So, yes, it's true. Bigger fibers produce more force. In any fiber type, that is true. But fibers of the same size, if they're different fiber type, the 2s will produce more force. And that's because there's something intrinsic about the sarcomere, either how it's binding or producing force, that's different. Okay? They're the same size, but something's going on inside of the fiber that's making it able to produce more force relative to the same size of fiber of a different one. Let's translate this back to the motor unit recruitment, right? We can't say this low threshold motor unit is this fiber type, but what do we know about motor unit recruitment and force production?
As you recruit more units, you get more force.
As you recruit more motor units, you are able to produce more force. Which motor units that are later are probably which fiber type? 2A and 2X. So if we're starting to think, okay, if we're recruiting more motor units, we're probably recruiting more fibers that are able to produce even more force relative to their size. So why do they produce more force relative to their size? On the exam, I will not ask you what is 2 pi b squared. I won't ask you that, okay? I won't ask you what constant b is or constant c is. I'll ask you, what does this quality mean, okay? So 2 pi b is essentially the transition time of actin myosin from weak to strong binding, right? So we remember the power stroke, right? Cross bridge cycling, right? Originally, you learned that they, power position, binding, pull, release of ATP, or rigor, release of ATP, new one goes on, hydrolysis, power position again, so on and so forth, right? Those are all the steps you learned. But realistically, there's probably about 10 to 15 more specific steps along the way. There's conformational changes of myosin and interaction ATP with the myosin and the actin. So originally, when myosin first binds to actin, it first binds in a very weakly bound state, and then it shifts and binds into a very strongly bound state, all right? So this 2 pi b says that the type 2 fibers spend less time, right, faster. They spend less time going from the weak position to the strong position. So that means that the type 2s, when they bind, they go weak, strong. very quickly. Whereas the type 1s, they bind and they stay in the weakly bound position longer, and then they get to the strong position. All right? So this means that they take longer to get into a strong force position. Yes, Mark?
That force position, are 2Bs going to be in like a better force position than 1?
It just means that they get to the stronger position faster.
But like for the strong position, it's like the same.
No, that's actually the next few right here. So, but good, yeah, we're going in the right direction. So this means that they get to the strongly bound position faster. And then if we look at T-on refers to the average time myosin is bound to actin is shorter. So this means that when myosin actually binds to actin, okay, it gets from the weak to strong binding position quicker, and then when it's bound, it does its power stroke and then releases, right? So its binding pattern is actually shorter in time compared to the other fiber types. So what does this mean for cross-bridge cycling?
You go faster.
They contract faster. Why do type 2s shorten faster than type 1s? Because they go from weak to strong binding faster, and once they get to the strongly bound state, they produce the force quickly and then detach, ready for the next cycle again. Whereas the type 1s, they take longer to get to the strongly bound position, and then once they get to strongly bound position, they take much longer to detach and go back to the power stroke, the reloaded state, right? So the shortening velocities happen slower, the cross-bridge cycling happens slower. If we look at these two, I won't ask you what is constant B and what is constant C. What they do represent, though, are when actin and myosin actually bind, right? This indicates, B indicates that actin and myosin binding is physically stronger, like meaning like I'm not just holding on to you to the binding, I am like handcuffed to you. We are in this together. It's strongly bound force production, right? We talked about like enzymes binding, right? Enzymes can bind kind of weakly and do their thing, or they can bind really tightly and the reaction happens fast and readily, right? This means that the type 2s bind very strongly together, actin and myosin. The type 1s, they bind, but not as strongly binding as the 2As and 2AX hybrids. So notice that anything that is not a pure 2A or 2AX is not producing the same amount of force relative to the other ones, right? So it's something about having any quality of a 1 myosin heavy chain is kind of like detrimental for you to produce force, okay? So the binding is stronger, but then C is talking about the stiffness of the sarcomere. So for example, here's my lanyard. If I push my hands together, is there force? No, right? Is this stiff? No, right? It's not stiff, so if I go to apply resistance, there's no resistance. My muscles, my hands can't develop force because there's no force to resist me because it's not very stiff. But this marker, is this very stiff? Right? So if I push against this, is there force development between my hands? Yeah, the harder I push, the more force I'm able to produce in this area because this is a very stiff structure. Type 2As and type 2AXs, the type 2s, they have stiffer sarcomeres. Imagine, think of a bridge, right? A bridge, if it's a good bridge, it's very stiff, very stable, very resistant to forces. A wobbly, those ladder bridges or the rope bridges, not very stiff. They're kind of like all over the place, right? Indiana Jones, look over, you all see that. Those ones. Anyway, they're not very stiff, right? So that bridge is not very stable because there's nothing stiff about the application of force. But if you have a concrete bridge, steel bridge, it can resist a lot of forces because they're very stiff. So think about the sarcomere and force production the same way. Because type 2s, because the actual sarcomere is stiffer, you're able to transmit force more effectively through the sarcomere, through the myofibril, through the muscle fiber, through the muscle better, right? So if you have a muscle that's more type 2 or you're producing more force from type 2s, it's going to produce more force compared to a muscle that's only type 1 because when the 2s are contracting, they're producing more force than the sarcomere, and also the binding at the sarcomere is significantly higher than the ones. All right? So, when we go back to motor unit recruitment, we say, oh, we recruit the low threshold motor units first. Why? Why do we recruit them first? Yes.
Is it a lower threshold?
Their lower threshold, because their lower threshold of forces are, they're low threshold because lower threshold of forces are required to turn them on, right? This is called a low threshold motor unit because it only required a low amount of force to turn it on. This would be a high threshold motor unit because it requires this much more force, much higher force to turn on. So if we think about force production in the motor units, if the lower threshold motor units in general are producing less force, it's because, well, they're innervating less fibers. Those fibers also happen to be smaller. But don't smaller motor units tend to be more oxidative? Yes. So they're probably more the type 1s. But guess what? Though you're turning on those smaller motor units, those low threshold motor units, they might be more oxidative, but they're not able to produce as much force. Why? Because even in the sarcomere, they're not binding as strong and their sarcomeres are not as stiff, right? They still develop force, but they don't develop force inside of the fiber to the same extent or as forcefully as the type 2 fibers, right? So which types of intensities are we more likely to recruit type 2s for? High intensities. It's high intensity because it probably requires higher
Force.
uses more carbohydrates, but it requires more force, right? Higher loads, higher intensities are higher because they're higher forces, higher loads, right? So we can't just turn on 2X's. We have to have a heavy enough load to stimulate those neurons to then turn on those motor units to recruit those fibers. And then those fibers are very big and they develop a lot of force, which is why those fibers are used in the higher load or high intensity exercises. Does that make sense? Right. So I know it's all like, I keep reiterating the same things, but this goes back to 346 when we're talking about the fiber type qualities, right? It's not just, okay, yes, this muscle fiber type has a lot of mitochondria. This muscle fiber type uses the electron transport chain or uses fats. Yes, that's true, but also notice like the force capabilities of that fiber type are not really well built to produce force, right? The sarcomere isn't producing as much force versus a type 2, type 2A, 2X. 2A can be more oxidative, but guess what? It's a 2, so it's probably going to produce a lot more force relative to its size compared to the same type 1 fiber that could have been recruited a few pounds ago or a few intensities before, right? So it's almost like the more force that's required during an exercise activity, the more you're able to recruit motor units and muscle fibers that also produce even more force than the ones before it. Does this make sense? Right? Okay. You'll get asked probably 2 or 3 questions about the sarcomere characteristics. So you've got to know that, yeah, muscle fibers of a certain size, yeah, larger fibers produce more force, but also what about the characteristics of those different fiber types? You should know, I won't ask you what is constant B or constant C, I'll ask you what about stiffness, what about binding, what about cross-bridge cycling, right? What about transition from weak to strong binding? Those would be the terms I use, not the actual I-squared thing. Yes?
What allows the type 2s to be more stiff?
It can be, well, one, it can be the binding of actin myosin, but also there's other structural proteins. So you have like the Z discs, right? Or there's an M line in the middle and the Z disc on the outside, but you have other proteins inside the sarcomere that are more structural in nature, like desmin, like titin. Those other proteins are supportive of the sarcomere, which also have some differences between fiber types as well. So yeah, like the stiffness as a whole can be because the organization of actin myosin, the density of the actin myosin, the titin, desmin, and other things that are also all part of that fiber type unit in that sarcomere, which translates to the fiber at large. But ultimately, it's that stuff. A multitude two different things, but kind of related to that, right? One thing we'll talk about here too, we just said that bigger fibers produce more force, but if you're a type 2, you're going to produce more force than a type 1 if you have the same size, right? But so there's something to be said about there being not just the fiber size that's important for the ability to produce force. This is a study that was done a few years ago, well, I guess almost 10 years ago now, I think 2015. I'm like, oh, it was almost 10 years ago. Anyway, they looked at the fiber size and the force production of different groups of athletes. So we have bodybuilders, power athletes, and controls, recreationally active controls. Bodybuilders are built to have a lot of muscle mass, so they're training to get their muscles larger. Do larger muscles produce more force? In general, yes. Power athletes also train for power, so they need to produce force, but very quickly. So the emphasis isn't so much on the muscle size, it's more about how we apply that force. And in some cases, you need to have that muscle be bigger to produce more force, right? So with power, it's more about how fast we apply the force. With hypertrophy and bodybuilders, it's just about getting it big. But ultimately, both those groups of athletes have larger muscle fibers and produce more force compared to recreationally active controls. So that makes sense. One group is doing lifting, the other group is not. But if you look at the force produced relative to the size of the muscle fiber, bodybuilders actually produce less force relative to their size compared to the untrained controls, right? So, and actually the power athletes like rugby players, football players, soccer players, they produce more force relative to their size even compared to bodybuilders, right? So this would suggest that maybe that there might be something called sarcoplasmic hypertrophy. Have we heard of that before? Right, so that would essentially mean, you know how you can look at the cross-sectional area of a muscle fiber, right? That's the area of the fiber. The perimeter is a sarcoplasm, right? So imagine sarcoplasmic hypertrophy would be that the fiber's gotten bigger, but hasn't gotten as strong relative to the change in size. So for example, when we talk, usually when we talk about muscle hypertrophy, we're thinking myofibrillar hypertrophy, right? Meaning that here's the original muscle fiber size. If it gets 2 times as big, twice as big, let's say you had 3 sarcomeres in here. All right, 3 sarcomeres. If it gets twice as big, and it gets proportionately as strong as it got as big, what makes it as strong? You gotta have 6. Right? So this is twice as big, but also twice as strong. What if you had a fiber that got 2 times as big, but three and 4. Is it as big as the other one? Yes. Would it be as strong? No, right? So now we're seeing this disconnect between muscle growth and fiber size. This is why you don't always see a 1-to-1 relationship between the exercise-induced change in size and the exercise-induced change in strength. We know that muscles can get stronger without getting bigger. But we also know muscles can get bigger without getting as strong as their size would suggest, right? A muscle twice as big doesn't always get twice as strong, right? Literally, they've shown supercompensation with glycogen, carb loading, makes muscles more full. And it has been shown to alter cross-sectional area of the whole muscle size just by doing resistance training and loading your diet with lots and lots of carbohydrates. Muscles will swell, right? The muscle didn't get any stronger, but certainly the area's gotten larger because it's swelled up, right? So that makes this relationship between the 1-to-1 relationship between size and strength starts to become less linear, right? In general, yes, there is still, in general, bigger muscles produce more force, but this is showing that there's some variation in how maybe that happens, okay? What about in this case where you have muscle, right, doesn't get as big. 3. Right, so this didn't get as big as the other one, but it's still producing as much force as this one. They've kind of hypothesized, they've kind of called that sarcomere packing, right? Because the sarcomeres are packed into a smaller space, right? So if you think about like a bodybuilder versus a powerlifter, remember last lecture it had bodybuilder, powerlifter, right? We said strength versus hypertrophy, right? The bodybuilder's main point is to get as big as possible, right? Not so much about getting as strong as possible. Yes, bodybuilders will be strong because they have large fibers, large muscles, but they're not specifically training for strength, right? So that means like maybe their muscles are not specifically adapted to produce as much force inside of it, despite the fact that they're very big, right? This is why who's seen where you've seen like really little people be really, really strong. Oh, you're a lot stronger for your size than you look. You've seen that before, right? Part of it could just be neurological. Part of it could be because maybe you have more type 2s and they're stronger fibers, as we know, relative to their size. Or maybe you have more densely packed muscle fibers, or sarcomere, potentially, right? I bring this up because they have shown data that when you do really, really high volume resistance exercise, they called it sarcomere or sarcomere dilution. So they had the study where these already trained people, like 5 or 10 years of training history, they did really, really high volume resistance training. And they saw that the muscle fibers got really big, but the concentration of actin and myosin in the fiber actually went down relative to the size of the increase in fiber. Right? They still got stronger, but if you look at the amount of actin and myosin in a certain amount of muscle that was analyzed, there was slightly less actin and myosin. And I'm like, well, they got bigger muscles though? How was there less actin and myosin? Maybe the fiber size got bigger, but they didn't add as much sarcomeres to their muscle relative to the increase in the size. So the sarcoplasm hypertrophied, but not necessarily myofibrillar hypertrophy. They repeated that study again, except this time they didn't do very, very high volume. Instead, they did really high load. And guess what happened? The concentration was consistent. The fibers got as big as they did strong. So then their hypothesis was, well, maybe there's something about high volume exercise that causes the fiber perimeters to get big, but not necessarily add the same amount of sarcomeres. So it could be the high volume nature that does that. They also suggested alternatively, maybe fibers need to get bigger first, then they add the sarcomeres, right? Maybe you gotta make space in the cell first, and then once the space has gotten larger, then we add the sarcomeres to fit in that space. Ultimately, we don't know, right? That's why they hypothesized. But I did a few studies to try to answer those questions, and it does look like, yeah, higher volume workouts do tend to make muscles bigger, but that doesn't necessarily always translate to the same increase in strength, right? Because obviously there's neurological adaptations, but even within the fiber, in the muscle itself, there's some data that suggests that doing more volume might make it bigger, but may not add the same amount of strength at the same time. So it depends. Yes?
Wouldn't that last statement contradict like your first diagram?
That they suggested that maybe the authors of that paper suggested that maybe there's a thing like sarcomere packing, right? They actually haven't shown sarcomere packing, but when you look at this, where they were still strong fibers, they weren't as big as the bodybuilders, but they were still really strong. But if you look at strength relative to size, it's actually more than these groups. So maybe it is packing, right? But it's not a very unanimous finding, and it kind of varies depending on the training protocol implemented, how we analyze the fibers, right? And the training status. Bodybuilders as well take all kinds of steroids, right? So is it possible that then taking anabolic steroids made the fibers so big and the fiber just grew, but didn't have the sarcomeres as much either, right? There's a variety of hypotheses, but ultimately they're just trying to explain why they found the results they saw. Doesn't mean that is what happened, but they tried to provide some physiology to support what might be going on, all right? So,
I have a question.
Yes.
You said that high volume resistance training led to sarcomere dilution, and what was it? You said that was it low volume but high load
Yes.
led to
Not that dilution. They didn't see a decrease in the protein concentrations relative to size, so they saw that it was consistent. There was no dilution there. However, they thought that maybe it was because it was the high volume. The other thing is that the high volume study, they did that, I believe, it was shorter in duration. So the high load study, I think, was 12 weeks, whereas the high volume study was maybe 8 weeks. So that's why they suggested, oh, maybe they just needed more time for the fiber to then add the sarcomeres. That was their suggestion. But it's not a unanimous finding, so I wouldn't say like sarcomere dilution happens for sure with high volume. I wouldn't say that just because you're doing high volume, that means that your muscle's not going to get as strong as it should. I think it kind of depends on the type of training you're doing, and then obviously other factors as well. But there is data to suggest that that might be a thing that happens, because we do see that muscles do not always get as big proportionally to however strong they are, right? And vice versa. We know that you can get strong without getting bigger. They've also shown you can get bigger without getting significantly stronger too. So it just kind of maybe depends on the training status, the type of training, the rest, all types of those things. But I guess I'm saying there are hypotheses about this certain style of training and maybe what implications it has for adaptation. So if I'm a strength coach, then maybe I might be a little more weary of super high volume training protocols, because maybe it's not going to give the best bang for my buck for my athletes, right? But if you're a bodybuilder or a physique athlete, it might mean, well, who cares, right? I should do the high volume because the higher volume in the training actually is related to more growth. So maybe that's more important. So it kind of just depends there. And then lastly, we see here with the fiber types that there's a range of oxidative activities in the type 2s and also their size. You see the type 2Bs generally are larger, but they have very low oxidative capacity because they have low succinate dehydrogenase activity. Whereas the type 1s, they are smaller, but they have higher succinate dehydrogenase activity. If you look at the 2As, they're kind of this bridge in the middle where you can have smaller 2As that have high oxidative capacity, and then you can have some larger 2As that have lower oxidative capacity, but they're larger, so they produce more force. So these 2As are really this bridge between these fibers you're using during aerobic versus anaerobic type of stimuli, and we're going to be able to recruit them. Some have more oxidative potential, some not as much, but they have certain benefits to each one. But that depends on the stimulus, right? And obviously this kind of all relates back to the fiber qualities you learned in 346. Type 2s handle contractions faster. They have better calcium handling speed. Type 1s, more lipid droplets, but they can use those lipids because they're highly oxidative. That's opposite from the fast glycolytic. Not a lot of oxygen, can't use it very well, so they use anaerobic metabolism, right? But also those anaerobic fibers are built for force and speed relative to their size. So kind of ties it all in there and how they're related to each other. Make sense? Right. So now we'll talk a little bit about fiber type shifting, okay? So if we look here, some adaptations to training, we already learned with cell signaling that when the muscle fiber contracts, you have the nervous system input. That action potential causes the release of calcium in the cell. When that calcium is released, so that nervous system is firing, when that calcium is released, it's turning on transcription factors that make the muscle more oxidative. We also know that when that calcium is released, it allows actin-myosin cross-bridging, which makes us use ATP, changes the energy status of the cell. Notice how frequently and how much that muscle fiber is firing, or the motor unit is firing or contracting, is dictating both the energy use and the calcium release inside that fiber. So how much energy we're using turns on PGC1 alpha, makes the fibers more oxidative. How frequently we're contracting turns on calcium signaling, turns on PGC1 alpha, makes the fibers more oxidative, right? But these are different pathways that are activated from an energetic or volume standpoint compared to heavy loading pathways. So resistance training is very heavy loading. Heavy loads activate mTOR more than light loads. But that activation of mTOR leads to more protein synthesis, which makes fibers bigger. And ultimately what we end up seeing is that you're not going to see a type of training that makes fibers really big and really oxidative at the same time, because there's competition between the pathways and the stimuli that make them bigger versus smaller, versus faster fiber type versus slower fiber type. When we look at this graph, we can see that the stimulus that's applied to the muscle influences the size of the muscle drastically. So for example, you could take insulin-like growth factor, which is a protein that makes muscles grow, growth factor. You could take beta agonists, right? Beta agonists are beta adrenergic receptor agonists. Where did we hear of the beta adrenergic receptor before? Where did we hear that?
Like epinephrine.
Epinephrine. So this is a compound you can take that stimulates the beta receptor similar to epinephrine. But guess what? It stimulates it in a way that causes the muscle fiber to grow. It's anabolic. So physique athletes will take a compound called clenbuterol because we know epinephrine causes lipolysis, right? Lipolysis gets rid of the fat. Normally to do that, you have to be on a diet with an exercise response. But if you take clenbuterol, it's stimulating the receptor like epinephrine without even doing exercise. So your lipolysis goes up 24/7. Problem is that clenbuterol is interacting with all the receptors in your body. So cardiac, heart rates are higher, blood pressure is higher. It causes the muscles to grow, but it also causes the heart to grow pathologically, right? So clenbuterol is illegal because, well, one, it's a legal substance. It does stimulate the muscle fibers and lipolysis and all that other stuff, but they've also shown that chronic use and abuse of those beta agonists like clenbuterol actually leads to cardiac events, right? Maladaptations in the heart. It's bad for your health, right? Because the heart muscle will grow. And if it grows pathologically, reduces stroke volumes, heart's working harder, the whole series of effects, right? But beta agonists cause muscle fibers to grow, and we know that androgens, remember, don't get beta adrenergic receptors confused with androgens. Adrenergic, androgen. They sound similar, right? But these adrenergic receptors are stimulating the epinephrine receptor. The androgens are stimulating the testosterone receptor, the androgen receptor, right? So testosterone is anabolic, yes? Beard growth, bone growth, muscle growth, right? To give any sex testosterone, they will get more muscular. They will develop male sex characteristics, right? One of them being is enhanced muscle growth, okay? So we know testosterone and androgens, androgenic anabolic steroids, cause muscles to grow. We also know that resistance training is a very potent stimulus for growth as well because it's overloading the muscle. So there's a variety of activities or things you can do to make the muscles grow, and it affects it drastically. So you could just, they've shown when you start running, type 1 muscle fibers will hypertrophy, right? Because you're overloading those muscles with the stimulus. But running is low load, so those ones do not get as big compared to if you were doing a lifting program, right? So yes, activity and loading makes them grow, but is not as effective at hypertrophy compared to actual resistance training or taking anabolic steroids. And this differs from these catabolic type of situations where disuse, right? Who's ever, you were less active for a few weeks, right? COVID, right? Lockdowns. People couldn't go to the gym anymore. I lost my gains. You were not as active, right? But when you trained again, they came back very quickly. Fasting. People who go on a diet, you lose weight. You lose fat mass, but you lose around 25% of his muscle mass because caloric deprivation. Cells need energy. One way they do it is gluconeogenesis. They break down the protein, turn it into a carbohydrate backbone, you can use that for energy. Unloading. Who's ever broken a bone and been casted? Right? You get a cast, take that cast off, fibers atrophied, muscles atrophied, right? Why? Because there's no loading, there's no stimulus to the muscle, right? So we know that when we're unloading or not activating the muscle, that also causes atrophy and also cortisol, right? Glucocorticoids. Who's ever had a cortisone shot? Cortisone reduces inflammation, but also it's catabolic, can break down muscle tissue as well. So if we think about how can we use this information, well, if someone is going to do a diet, which is going to cause us to lose muscle mass, we can also incorporate a resistance training program so that we're not losing the muscle, right? It stays the same size. So we can counteract these catabolic effects by introducing an anabolic stimulus. They used to say, oh, well, steroids, you have to go to the gym to take steroids too. Steroids don't just work. You can't be a couch potato and just take steroids. In 1999, 2000, they did a study out of UMass Boston with Boston Medical Center, and they had 4 groups. A group that did steroids only, no exercise. A group that did exercise and steroids, exercise only, or nothing at all, right? Just control. The group that took steroids and no exercise gained as much muscle size as the group that exercised, right? They didn't get as strong as the group that exercised, because remember, part of it is neurological, but they did get stronger because their muscles grew. So that whole concept of you can't just take steroids, steroids only work if you lift weights, that's disproven. Steroids will make you bigger and stronger just by taking them and doing no exercise at all. Equivalent to someone who's exercising. Now obviously that's in the untrained state. If you're resistance trained, probably wouldn't see the same effect, but at that point, then the effect is, if you're resistance training and taking anabolic steroids, you get the best effects, right? But it comes with deleterious health effects, too, right? So unhealthy change in physiology. But the idea is that whereas things get bigger from these stimuli, that also causes them to become a slower phenotype. So we can see that the fibers, once we start activating them, they start to shift to become a more oxidative, slower phenotype. If we're not activating those fibers, they become more fast phenotype. So like the 2X's, when you're untrained, you're not stimulating them. When you start training them, they shift to become a 2A. And that fiber type is better at doing more sustained contractions. If you stop training those fibers, those 2As will shift back to a 2X because there's nothing stimulating them to maintain that oxidative phenotype. So they shift back to the 2X because there's no stimulus to keep them that slower phenotype, right? So the 2X's will shift from 2X to 2A and back depending on your training activities. But you're not going to see a shift between a 1 and a 2X because the neurons are very different from each other, and there is no shift in between the fiber types in that way. But ultimately, as why we don't see really big fibers and really oxidative fibers, because the stimulus, one stimulus is causing fibers to get really big and really big and more anaerobic, more faster type, and then the other is telling fibers to stay smaller but become more oxidative. Right? Smaller fibers are better for oxidative. So we're not going to see both of these at the same time, which relates to that the more training we do, the more specific our training needs to become, okay, to see continued adaptations. Now obviously we have fiber types. You know, here's a pure 1 fiber. Here's a hybrid fiber. It has heavy chain 2A and type 1. Here's a pure 2A fiber. Here's a hybrid 2A, 2X, a pure 2X. This is a control lane that just tells us which band is which fiber type. The lowest bands are the 1s, and the 2As and the 2Xs. But what we end up seeing with the hybrid fibers is that these hybrid fibers, they're about 10% of your total muscle fiber type. But guess what? They tend to shift towards the type of training you're doing. So if you're doing training that is more anaerobic, more high intensity, this type 1, 2A hybrid will become more 2A. If you're doing training that's more aerobic, more long distance, this hybrid will become more type 1. And this 2AX will become more just 2A. So these hybrids will shift to help support the type of training that you're doing. But you're not going to see a shift from a 1 to a 2X because this 1 fiber is innervated by a small, low threshold motor neuron, right? that neuron itself is not going to change. The nerve will not then suddenly become innervated by a type 2. That doesn't happen, right? So that only see where we're seeing changes in the fiber type of the neuron is through aging, where we've lost the neuron, or then it's innervated by a type 1, right? We lose those high thresholds, so those are the case. But we do know that changes in fiber type happen relatively quickly. We know that adaptations in training happen very quickly. So here they had done repeated testing in men and women for I think 16 weeks, and they saw increases in 1RM in as little as 2 weeks of training. Now we know that part of that is neurological, right? We've already seen that in the last few lectures. But what we also know is that shifts in fiber type also happen very quickly. In the women, they saw the type 2X fibers go from 21% to 12% in just 2 weeks. So you halved the amount of 2X fibers in as little as 2 weeks of training. For the men, it took up to 5 weeks, then they saw the shift significantly, right? But the idea here is that the shift in fiber type actually happens relatively quickly, depending on the volume and the stimulus of the training. We also know that if you're lifting and doing anaerobic exercise, that is going to make the fibers become more anaerobic. Whereas just because you're a trained endurance athlete, that doesn't make your fibers significantly stronger or better at in an anaerobic type work. Essentially, if you look at rates of force development, cross-sectional area, muscle fiber conduction velocity, whether you were a power athlete or a strength athlete, both of those anaerobic groups were better than sedentary and endurance athletes. And essentially, the sedentary and endurance were the same for all of these qualities. So if you're an endurance athlete, and you might be a great endurance runner, but if you compare your physiology compared to a strength athlete, you're essentially an untrained person, physiologically, right? If you're comparing those two groups. In anaerobic capacities, anaerobic works, right? You're still trained, but you're not anaerobically trained, and that's where, you know, your specificity of training comes into play. Okay. Now, in this study, they had power lifters, and they notice that we have the controls and the well-trained lifters. Notice how the highly trained power lifters, they show almost no 2X's here, right? They did also do the cross-sectional areas, and there were 2X's there, but most of the 2X's had shifted to the 2A. Notice how only one of these lifters actually has a third band where the 2X's are. Almost all of them are just 2A's. Whereas the untrained people, they're not used to stimulating those high threshold motor units, so of course they have 2X's because they never use them. They've never shifted or converted to the 2A's. So untrained people have more 2X's. Trained people or people who do training have less X's and more A's because they shift to that fiber type, because the stimulus, the contraction, tells them to shift. Now, how do we shift that fiber type? From resistance training, they had two stimuli. One was 60% 1RM, one was 80% 1RM, and they looked at the mRNA, the gene expression post-exercise between the three fiber types. We can see that in the type 1 mRNA and the type 2A mRNA increased up to 6 hours post-exercise. Right, so for the 2A, it was increased post, 2 hours post, and 6 hours post. But notice, the 2X mRNA only increased up to 2 hours, not up to 6 hours. So you use that mRNA to produce a protein, correct?
Yeah.
If one mRNA is produced 4 times as long, does that mean you might produce more of that protein? So that means that you're still producing the X, but over time, you're going to be producing more of the A and less of the X compared to each other, okay? And it didn't matter whether you did a 60% or an 80%, you were producing more mRNA of the 2A's and not as much of the 2X's. When we look at within that individual, I love this study, they had the same individual, but the one leg did 2 workouts to failure, the other leg in the same individual was just control, no exercise. Every single one of these white fibers is a muscle fiber that is expressing 2X mRNA. So this means that this is a 2X fiber. Why? Because it's expressing the mRNA, the gene expression of a 2X muscle fiber, right? It's showing up as white here. They did one workout, and what do we know? 24 hours after that workout, what happens to the intensity of that 2X gene expression?
Goes down.
It goes down. They do another workout, again, leg press, knee extension, 4 sets to failure, 2 exercises. One workout, then 2 days later, another workout. What's the difference between the control leg and the work leg?
One's working. Oh, no, two.
The leg that was doing repeated exercise actually is producing less 2X mRNA. If you're not producing the mRNA, how can you make the protein?
Yeah.
You don't, but don't we still produce 2A? So even though these fibers aren't producing 2X mRNA, they are still producing 2A. So these individual white fibers that are 2X, they will now become a 2A. So the X's convert into 2A's because we stop producing the X protein, but we do produce the 2A protein. Over time, those X's convert to A's. And remember, if you stop stimulating those fibers, you start producing the X mRNA again. So then those A's will go back to the X over time. Now the thing is, you might think, well, don't we want our X's? They're high force producing, they're fast contracting velocities. True, but if we're lifting loads frequently enough that we're recruiting those fibers all the time, don't we want to be more fatigue resistant? So if we're going to do that type of exercise to failure more often, it would be better if our fibers were 2A because we can produce more force repeatedly versus a lot of force, just a little bit, and then fatigue, right? The 2X's can contract very fast, but that comes at a cost of them fatiguing quickly. The 2A's don't contract as fast, but they can produce a lot of force for a longer period. And if you're doing steps to failure repeatedly, that's the type of training you're doing. Lots of volume to failure. Yes, Anna?
If you had worked out and then stopped and gone back to working out resistance training, would the transition from 2X to 2A be quicker, or does that happen at the same rate?
I think it still happens at the same rate. We know for sure, which we'll cover with muscle memory, the size comes back twice as fast, right? So the muscle size and strength returns twice as fast versus if you had never trained at all. But I think the changes in the fiber type happen particularly the type 2s, they tend to happen at a similar rate, but it does happen very quickly. In this case, we saw the first study was, 2 weeks we saw fiber type. Here we saw in as little as 4 days, 96 hours, and there was a significant drop in the 2X fibers from doing just 2 workouts, 2 extras, 4 sets to failure. Very, very intense workout, realistically. But it happened in just 2 workouts. But that's meaningful, right? Because if I'm a power athlete, that means I don't want to be doing high volume work. Because guess what? I'm losing the X's, which are velocity. I need those for power, right? So we'll finish the rest of this next class. We'll cover satellite cells and ribosomes, maybe some muscle memory, we'll see. But we're still shooting for the exam to be on the day it's supposed to be. So unless, if there is a date change, I will tell you that next week. But for right now, we are planning to still have the exam on that same day.